It's going to be your camp some. And hey, remote people, just in case you're wondering, we're still waiting for people to come into the room. Okay. Yeah. At least Florian appears to know about it. Yes? It's the entire screen, as far as I'm aware.
How do we even get our options?
Sorry?
How do we get our options?
So I have. Let me just close that. Oh, sorry. Recursion. Sorry, I have the presentation thing open. Presentations thing, and so if you could post. Sorry? Oh, your thing is a. Oh yeah, sure.
I'll just make it simple.
Cool. No.
My macro.
Yeah. I know, I'm like, no. Oh, yeah. Okay. It looks like everyone's here, I think. One, two. Emil's, Karen. Okay. We're good to go. Okay. I guess we will start. So before we go to presentations, I just want to make a few very short remarks. Firstly, you guys are all awesome, obviously. I've been so impressed with all the work you guys have done. As I told a few of you, I feel like I've achieved nothing compared to any of you. Every day I sit down and I'm like, I'll get some other stuff done while I'm like organizing lunch and stuff, and then nothing happens. So amazing to see what you guys have achieved. First of all, I'm going to go through the sponsors because they have been very useful. So Ecoscope being our biggest sponsor, helping us with the venue and kicking in some money as well. So thank you very much to them. Our Megabase sponsors, Genome BC, Michael Smith Foundation for Health Research, and ACM Women's Chapter. Also, very big thanks to them. Our nucleotide sponsors, Artheo, NumFocus, Chipotle, New England Biolabs, and the Bioinformatics, Canadian Bioinformatics Workshops, or bioinformatics.ca. Thank you very much to them. And our community partners who have kind of helped us spread the word about this event, NCBI, GrassPods, the Society for Canadian Women in Science and Technology, VanBug, Software Carpentry, and Challenge Rocket. So thank you. And lastly, a big thank you to all the hard work of people from ORCA at the BC Cancer Agency and Compute Canada for helping us get reliable compute infrastructure. Cool. And thank you to all the volunteers. So a big hand for sponsors and volunteers, please. And lastly, for all of you, fame and fortune awaits. And some of you sound like you're really close to getting a paper done, which would just be amazing. So just keep pushing it. I'm sure there's still more edits that you want to do, but let's try and get some of them online soon. Cool, and that's it. Let's invite the first team up, Artem's team, to have a nice chat. I guess I have to enable editing probably. Cool.
Hello, everyone. My name is Artem.
My name is Gavin.
And
I'm Jasper.
And then we also had Anisette working remotely with us from the Ivory Coast, and Jeffrey all the way in Ontario. And then Alyssa and Eric couldn't make it today. So our team is Biosyntax, and our goal is to make biology more human readable. So the problem which I'm sure everyone in this room has faced is you need to figure out what this read is, and then you're given a bunch of data, and how do you figure out what's important here, and how do you actually go about reading this? So we're all probably familiar with this idea of syntax highlighting, and that's that you recognize classes of text, and then you assign colors and formatting to them. And this is a way of organizing information. So the question is, in biology, can we make computational biology more intuitive to the users?
So we try to target file formats that most bioinformaticians have faced. Okay. All right, very good. So we try to target file formats that most biologists have faced before, such as FASTA, FASTQ, SAMS. We added the ... in the end because we probably want to integrate more if we have time in the future. But essentially, the idea is how can we make these files more readable and interpretable for biologists when they look at it in general. So, for example, the easiest one, the FASTA file. When you first look at it, you just see a bunch of black and white text essentially. But by adding a simple overlay across it over files and text, we're able to see the alignments a lot more clearly. And if you try to expand it across a larger data set, when you first take an initial look at it, you can clearly see where the alignments are, which makes it a lot more elegant. Essentially, we did the same thing with FASTQ, and we created a gradient scale for Phred scores, so you can see where the high quality and low quality rates are, and low quality bases are. We also added a gradient scale to rate files, so I mean, you could essentially see areas of, I guess, large coverage. And the important factor here is that we try to unify the same content across different formats. So, for example, genomic coordinates would be the same color across different files. So when you first open the file, you can clearly see what it is you're trying to find per file.
Yeah, and VCF is an example of complicated file where syntax highlighting actually helps you to parse the data quickly and find the information you are looking for, make it more understandable. And actually, PDB is another example. In the header section, you can highlight valuable information, for example, authors or title of the paper where the structure was published. But in my opinion, more important is that you can actually highlight some data information. And when user wants to, I don't know, find something really valuable, for example, structure, resolution of the structure, he can just look into the file and find relevant information very quickly because it's highlighted.
So SAM is one of the probably most heavily used ones, and again, this is where I was struggling, where you're going through SAM files and just actually reading these end up being really difficult. So one area we really focused on was making sure that the headers can be very pretty. So like, as you can see in blue is a lot of the kind of libraries where these are from. The reds in the header are the technology. The orange is the command line. And then we also applied a lot of the things that we learned in the other file formats to SAM. And so again, you can see in green, here's a bunch of coordinates. But there are really interesting properties which began to emerge where because everything is sorted by the alignment, right? You can actually see the alignment in the SAM file directly. So when you're opening it and going through it, everything becomes really, really intuitive. You see the reads kind of along the genome almost. You also can identify features of your alignments, such as if you have a lot of PCR duplicates, you just get blocks where all the colors are all the same. And then again, as you can see on the side here, the Fred scores, these regions that are dark, this is just areas where the Fred score is just not so good. So right away, like, you're going from reading a black and white text where you have to kind of really squint your eyes and get everything to the data just pops right out of the screen at you. So we've currently focused on a text editor called Sublime, and this is available for all types of computers. And we're now porting this into Vim and gedit. So we've done a few of the syntaxes, some of the simpler ones, but over the next kind of maybe few weeks, we'd like to finish these 3. That way, this syntax highlighting will be available to everyone depending on whatever text editor you choose. And then the big advantage of something like Vim is that if you can use the terminal, that means when you're logging into the GSC and spot checking your files, they'll automatically be highlighted for you in the terminal. So just to summarize, the main kind of points of biosyntax, why you should download our repo is you're going to interpret your data very intuitively. You're going to do this way faster with less errors. And that means that in the end, it's just going to make you a better biologist. Thank you.
Let's have a few questions.
Do you plan to account for people who are red, green, line?
Color block?
People who can distinguish red and green. Do you plan to?
Yeah, so we have like themes and like you can use any kind of custom theme, but we can change the themes. And the idea isn't so much about the particular colors that we're showing. This is just one theme that we are working on. It's the regular expressions together. That's the engine of biosyntax, right? That the output or how pretty or not pretty you think it is really, and it's pretty. So yeah, we will account for that in the future.
So for the case of like sound, like can you have this work with something like the BAM file, like where you stream, and as you stream while opening the file work?
Do you want to answer that? You're working with Vim.
Well, for the most part, when you're on the terminal, like if you just open it in Vim, it'll automatically like highlight the text based on the regex expression. So yeah.
So would it do like decompressing from BAM to SAM, and then you see it in Vim?
Yeah, as long as you load it into Vim, it'll work.
So SAM tools is going to do the decompression, right? Like you're going to use SAM tools view H or like.
But my just worry is that like, do you have to like decompress the whole file and then view it in SAM?
No, so it's whatever's in the buffer. So like whatever's going to be on the terminal, whatever's on your screen, that's where your regex is acting.
I have a question. Yeah. So I'm not quite sure how to phrase this, but like there's a problem of opening large SAM files with them because you can't load the entire file to memory if it's a large SAM file. So is there a way to do it in bus or something? I'm not sure. It's kind of my idea.
Yeah. We well, I don't know. Yeah, this is the one unfortunate thing about less is like it might not be feasible in less because we could probably write an actual like less module or something to make that a thing. But again, it's really going to be the headers and or put it to a file or put it right to Vim and use the head command, head and 1000, and then.
Cool. All right. Yes. Thank you guys.
Can we see your BCF? Cool. Nice.
All right. Kieran's team want to come up?
Right. Hello, everyone. I'm Kieran. I was the team lead for this. Hide? And what does that do?
Oh, sorry.
Oh.
Well, you don't need to, but down there.
Yes. Right. Awesome. Okay, right. So what we were setting out to do is to work further on something that I've actually built in a previous hackathon and I've had a summer student work a little bit on, and that's SCREW, which stands for Single Cell Reproducible Epigenomics Workflow. And the goal for that is to create take a bunch of existing methods, wrap them up into a reproducible form that other people can use, download a bunch of public data sets and make pretty pictures like this one here, which is not our picture, although I have actually personally kind of come close to reproducing this out of their data in other work that I've done myself, but we didn't quite get to that point in this project. Reproducible research. So in bioinformatics, we have this huge problem where mostly you just get the methods section in a paper and they say, we did this, 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 and this, and then it's actually horribly inadequate to reproduce exactly what they did. You don't have the software versions they used, you don't have the exact code that they used. And a bunch of people are kind of working on methods of providing reproducible kind of analyses. Common workflow language is a big one that's being done. Integrates with Docker over here. Docker is really neat because you can put all of the software under the exact OS you used. Common workflow gives you an exact description of what was done. And the goal with SCREW is to put a bunch of CWL wrappers and provide a Docker container that can do these kind of standard analyses. Common workflow language, as I said, is kind of a neat thing. It's like a nice general standard. It's because it's a standard and not kind of just its own language. It's actually supported by half a dozen workflow engines already, and like there are quite a few that are still being working on support for it, including Galaxy and Taverna, which is really neat. The problem is that it's also quite new. There's kind of a fairly small team of people working on it. It's not super well documented. And this kind of led to a lot of headaches for us. In fact, we're kind of, in terms of our actual hackathon experience, we didn't even really have a fully functioning dev environment by the end of the hackathon on anyone's machine, which was kind of a pain. One of the problems is that we have the Docker files inside the Git repo. When you branch that, the source code is also getting packaged into the Docker file, but it's like, it's being checked out of master because the Docker file tells it to check it out of master, and you can't actually inject the branch into the Docker file at build time on key.io where we're storing the Docker files. Or maybe we can, but it's definitely not something that's in the scope of solving in this hackathon. Then we're trying to run things without the Docker thing, and then the CWL tool was having issues with our environments, and various other kind of issues we were having. But like some of these issues we were grappling with, especially like CWL itself, like trying to run a single script across a bunch of files, which you would kind of think was easy, but it was actually kind of tricky and very poorly documented, but we did manage to get that running on a few kind of like sort of small examples. Just being able to handle passing lots of files around within CWL. CWL does a lot of magic where it's injecting files into Docker containers and things. So it is a little bit kind of tricky. And also figuring out how to get all those files into a useful place at the end. We literally, like Brian and I spent the better part of 2 days banging our heads against these problems and mostly solved them, but like we don't really have a lot to show for that in terms of like images or anything, but actually we have potential solutions that are like most of the way done that will be really useful in future as I carry on working on this workflow. So yeah, so we did manage to finish some stuff. And we also managed to mostly get, so there has been a kind of an existing workflow doing a few things like generating heat maps and things, which was kind of in hackish kind of half bash. And I'd presented some results briefly at like ISMB, but like literally it wasn't actually in full CWL formats. We've got to the point where that's almost done. We just actually need to have a dev environment set up so we can fully test it. Also, we managed to pull down all the data sets I was wanting to get down. Our remote participant did all that. So there's a little bit of curation needs to be done, but that's made a big kind of step towards actually putting those all in one place, because the goal at the end is to release one big unified analysis with the Docker file, all of the files available in one place, probably hosted by GigaScience, because they were kind of interested in trying to host it all as a kind of sample workflow. And also we got into figuring out rendering our markdown documents from an R script wrapped in a CWL wrapper, which itself is its own little like technical thing, but it's also a big deal because a lot of the final outputs out of this pipeline are going to be R markdown documents with nice rendered images and reports. So we learned a lot, and this is just, so Connor had been working on one of those kind of report visualizations, which is just to use GVers within the bioconductor GVers package to load up regions of interest, gene models and things, and then a bunch of DNA methylation files, and plot the average plot the methylation per cell across a region of interest, which would be calculated from other parts of the workflow. And so we did at least get to the point of plotting that, although we don't actually have that like wrapped up in CWL yet. But that's just to show you a figure of something that actually did happen in this workflow. And that's this hackathon, and that's pretty much it that I have to show for you.
Great.
Questions? Yeah.
So is CWL, like, did you define it in a file, like a Docker file?
Yeah, no, so CWL, you put a reference to the Docker file inside. So CWL works that there are mostly tool descriptions which wrap a single kind of tool. You put in a hint that tells it, use this Docker file to when you run this tool, and it goes and pulls that Docker file down and then runs everything. It injects it into the Docker image and then brings back results to you afterwards. And then CWL also lets you make workflows by chaining tools together, and has a few, and also to provide YAML descriptions of a particular instance of running a workflow. So the exact data that you pass through when you ran your generic workflow that you built.
I'll ask about GigaScience. That's not something
You haven't heard of GigaScience? They're like a journal that's just there to publish huge data sets. And they're like based in Hong Kong, I think. They're pretty supportive of open source and open access. And one of the editors came up to me at ISMB and said like, oh, I saw your talk, like, do you want to publish your final thing with us as a kind of GigaScience like data sets? Here we have a curated data set of all public data, single cell DNA methylation data sets with a fully reproducible workflow with Docker images and everything. And, you know, so that would be pretty exciting if I have enough time to get it all finished up and stuff. Right.
I guess quickly, so I've gone down the rabbit holes of like a bunch of different workflow managers online. I see there's like 40 different ones at the time.
Yeah.
So it's interesting to hear you say like, you know, CWL has like a lot of buy-in, and it seems like very well filed, but it's interesting, it's like tricky for you guys to get working. So do you think it's kind of like the way that it's designed is kind of a roadblock, or it's just kind of like the documentation and like the support and the documentation that's right now?
Yeah, I mean, I think both are roadblocks. Like the way it's designed is overly complicated by necessity because it's meant to be a generic standard that can be implemented by a lot of systems. I think Sean, I was chatting to Sean about this, and Sean was saying like he writes make files and then uses a tool that spits out CWL from make files now. a way of kind of generating CWL rather than actually hacking it by hand. And I know one of the, Seven Bridges Genomics has been funding the developer of CWL, and they've also got their own dev team building tools. One of the tools they've built is like a visual editor to try and make building workflows a little bit easier. But yeah, and wait, what is the other half of that question?
Yeah, I think, yeah, is it design issue or just like documentation issue?
Oh yeah, and it's also a documentation issue. I was kind of meditating that what I feel like doing if I have a free day at some point is making a minimal tutorial for CWL that does things like show you how to take all the output and put it into output directories, show you how to iterate over files, because that's stuff that is very poorly documented at the moment. And I literally had to ask Michael Crusoe, who's the editor, the developer of CWL, personally, like via GitHub and places, and he was linking me to example code, but it was all like deep, like some EBI metagenomics pipeline. So it would be nice to have just some minimal working examples, because these feel like standard things that people should need to be doing, but I guess kind of maybe aren't thinking of too much, or maybe the people who are developing it are kind of experts in workflow development and kind of just don't think of this stuff as being hard. But yeah, documentation is an issue too.
Cool, that's fine. Kieran?
Three. Okay, thank you everyone. I'm Rod. Joining me is Will, and Erica is hiding back there. But our project that we worked on is called FuseBench. The idea is kind of a workbench to help people work with genome fusions, mostly in a personalized medicine context. So I'll start out with the acknowledgements, the team, which was super helpful. So here we had myself, Erica, Will, and Alex, who is off writing our paper, I think and hope. Remotely, we had some great collaborators. We had Steph from the UK helping us out. We had Harak from Long Island and Samir from Chicago all helping us out, and they were great. They took on pull requests. They worked when we were sleeping, and it was a really fun experience. So just as a general background, I'm going to go quickly through this, but the slides will be available. So I'm really interested in gene fusions. I studied leukemia, and a lot of the subtypes of leukemia are really defined by these recurrent gene fusions. But when you look at next generation sequencing data, it's kind of a classic informatics problem where there's 20 different tools, all have different file formats, and people who have tried to put those tools together, and you see these like giant Venn diagrams, and the intersection is not pretty. Like all these tools kind of have different file formats, different outputs, and they don't really agree on what fusions are there. So for the sensitivity is generally quite good, but specificity is quite poor for people who think in those terms. So just for people very quickly, a gene fusion is literally, you have two chromosomes that are breaking apart, fusing back together. In cancers, those can literally fuse exons to make a fusion protein that'll have different properties. It might be expressed in the wrong place and can be a driver linked to cancer. We have all these callers, and we have results from all these callers, but they're all in different file formats. So kind of this big cloud here, hack, hack, hack. The idea is let's at least try to make these things like have the same file format as a first step to being able to compare these things to each other. So beyond that, the longer term direction for the project was once you have all these things kind of in the same file format, then you can start doing more interesting intersections, overlaps, make more exploratory plots, summary statistics, and actually kind of get to this point where you can do a better job of interpreting your fusions when you have them. So this was kind of the idea that we were getting through, working through. I don't know if we made it to the profit step, but that was the plan. Most of the other stuff happened. So one of the big successes of this project was we wrote a lot of parsers, and actually I shouldn't say we because I didn't write any of them. The team did everything while I was kind of laying down the tracks. So we have now fusion parsers for 9 different tools, and so these were all, some of them were kind of similar, but many of them were quite different from each other in terms of how they talked about scores, how they talked about gene fusions, how they talked about orientations. So that was actually quite a challenge. I'll talk a little bit about some of the issues we faced. The other useful thing that was not present right at the start was that there's a lot of databases for fusions. Those also all have different formats, all have contradictory information in them. So we also have parsers now to take these databases and turn those into a common format. So you can see on the right here, this was kind of like the end of yesterday, just everything merging in, and we managed to not have any huge Git catastrophes, which was a minor success in itself. So just briefly, just for some of the main actual informatics challenges, so even getting to the point of like defining what the intervals are, whether they're zero-based or one-based, closed or open intervals, was challenging to find. So VCF at least has what the quality score is supposed to be, like what the scale is on, but that is also not kind of the case for gene fusions. There's kind of this good and bad thing where the format we used, BedPE, which is kind of a paired end extension of bed files. So it does allow unlimited columns, which is great because different tools can report different things, but that also makes it difficult when you're trying to merge and compare things. A lot of the time, coordinate information was unavailable, so we had Steph heroically like back converting from HGVS coordinates and transcript coordinates into genome coordinates. Huge mix of gene names, transcript IDs, genome builds, so all kind of the usual bioinformatics problems, but that we actually managed to get through quite a few of. So this was kind of the revised workflow where we were trying to get to the point where you get everything down to a common format, and then you start doing interesting intersections to answer questions like, of the gene fusions in my sample, which ones are known in databases, which ones are intersecting between tools, which ones are not. So we got to this next step where one does not simply merge these files. So just the challenge that we're looking at, so if you have a single bed file that is a single set of intervals, there's a well-defined suite of tools. So bed tools is the most popular one for doing like intersecting intervals, but finding merging intervals between tools, but that's not actually that well developed for these paired bed files. So I'll just go through a couple of the problems that we faced. So there's all kind of the usual bioinformatics problems where, you know, chromosomes are not named the same thing between samples, which is, you know, that's fixable. But then even the order for 1 versus 2 is not really maintained. It's almost arbitrary for which is the first and which is the second. That's solvable too, but not something that we solved. Again, we have this like 0 versus 1 based encoding, or literally just different tools calling things different things. We have all the usual gene name problems. So MLL is a gene where the whole field still calls it MLL, but the real name is now KMT2A. So depending on which annotation set your tool uses, you get different names. And tools don't really agree on what strandedness seems to mean either. So these are all the issues that you have to kind of take into account when you're trying to merge these things. So we did get some part of the way. So bed tools is a great tool. It has lots of tools for doing this kind of intersection. The paired end tools has some of them, but they're not quite as mature as the single end ones. So it almost worked, but not quite worked. And Will did a ton of work doing that. So we're bashing his head against that. So I tried kind of the R side, so dplyr doesn't quite work. Data.table, Bruno helped me look for something, but that also didn't quite work for paired intervals. It works for single intervals again. So we kind of think that, you know, genomic ranges and things like that would be suitable. Right now it'd be kind of a custom tool. So this is not implemented yet. There is a pair-to-pair tool that gets you most of the way though. So Erica worked on some cool visualizations, which are kind of fake data right now, but this is kind of where we're aiming to get to, is to be able to say, like, what is the intersection between your tools? So we want to make cool upset plots. If you haven't seen these before, this is trying to show intersection between, in this case, 5 different databases, what that would look like. And then trying to do that between, like, you know, if you have potentially 7 different tools. So how do you do that? So that's what we're working towards, is being able to do this kind of intersection analysis with these disparate tools. So just to wrap up, so we had a really good time with Git. We managed to close a lot of pull requests and not step on each other's toes too much. Yeah, so in summary, I think the main contribution right now is just having these parsers and having that kind of intellectual effort to actually figure out what all these files are doing and what they're telling us. So that's actually going to be really useful for me, for my project. And pushing this forward is just doing the continued work to actually make this merging actually make sense, and then being able to do the downstream visualization and interpretation. So that's it. So thank you very much. The organizers.
You need a hug. Are you okay?
I'm okay. My brain kind of checked out at about 2 o'clock today, and I said, okay, that's enough.
I mean, so a lot of kind of snip calling pipelines these days, like GSC ones, can use them on a bunch of tools and then like intersection using what they want to trust-ish.
Yeah.
Is that kind of how you foresee?
Yeah. Yeah. So for example, like when I'm calling snips, I might use like 3 different tools and take an ensemble, whether it's like 2 out of 3 or like some kind of classifier. So other people have built classifiers that combine different fusion callers. We were wanting something a little bit more generic because a lot of the time when people build the classifiers, it's also kind of an internal file format too. So that's the idea.
About the idea when you look at 2 different fusions, if there's some sort of like window of considered the same fusion, or whether or not you're matching things like exactly based on coordinates?
Yeah, so we were kind of inspired by Bed Tool's idea of like, you know, allowing some slop. So like a lot of times like things will be off by 1 or 5 base pairs, and like there's going to be some kind of arbitrariness because like if it's 2 different isoforms where the exon boundary is like 5 or 10 bases, we would like to know that those are 2 different things. But a lot of the time, like one tool will report the last base retained, and the other tool will report the first base into the gap. So yeah, you have to kind of be pragmatic and merge things there.
We try to retain consensus sequences too, that gives you the great sequence.
So we were trying to get down to, so Bed defines kind of, or Bed PE defines like 8 core fields, and we're trying to be really strict about the core fields and really flexible about keeping everything else. Yeah. Great, thank you.
Team 4?
One person. All right, hi everyone. So we're team 4. We were wanting to make an interactive visualization to explore all these kind of diversity metrics that you can calculate for any community. In our case, it'd be more microbial community. So here's, I guess, our teammates. I'm joined with Ali right here, and I have some teammates in the back, Reva and Akif, and then Roshan stepped out for a bit, and then I had 2 remote participants as well. So just a little background. So, I mean, I mentioned a little bit about the bacteria that we're interested in, and we live in the microbial world. So there's bacteria that affect kind of a lot of functions in these kind of environments, like the coral reef. There might be like bacteria that interact that kind of talk about or explain maybe the health of the coral. There's, you know, the forest, and also like the bacteria on us. So ecologists might be interested in like the diversity of these communities, and the question is, why would you want to do that? And that is to, I guess, the general assumption for diversity, or the kind of number of species or different bacteria that exist in a community, is that the greater amount of diversity means that it's probably overall healthier, the environment. So in like the colons of our guts, or maybe in the coral reef. And then if you have a lower diversity of bacteria or less number of species, then the environment might not be as healthy as others. So let's see, what is it? Okay, so the motivation for this project is that ecologists have been studying this for quite a while, I think decades now. And you have so many of these kind of metrics to calculate on, and they have nuances in their mathematical calculations. So in, say, like an application like R, you can easily just pick and choose from like all these, I don't know, like at least 10 different metrics for these diversity metrics, maybe more. And then it's like, you just choose the one that makes your data look better or not. So we wanted to try to, I guess, enlighten, or try to have this interaction of, you know, kind of pick and choose, but like also try to inform like what these kind of calculations might do to your data, so that down the line you might choose one that's best for your data. So there's 2 types of diversity metrics. There's one called alpha and then one called beta diversity. Alpha diversity looks at the number of different species or taxa or kind of groupings, while the beta diversity kind of looks at if you have multiple samples and maybe comparing them. So it's saying like humans or something, and maybe you get like a gut sample from like somebody with like C. diff versus a healthy patient. So you might want to compare the two and see if they're the diversity of their bacteria is different. So that's one thing. So yeah, so the question is like maybe how will your data affect these calculations? Because here's some of them. They're kind of mathy. I'm not going to go into them, but some of them weight on different things like one important thing is maybe if a species appears only once in your sample, that might indicate that you haven't sampled enough. So if there's a lot of bacteria that show up only once, then that sample may, for some calculations, will say that it's more diverse. So that's just one example. So yeah, I mentioned just at the very beginning, so we want to make an interactive visualization. So we want to make use of R and then Shiny, a package produced by RStudio to try to make it really interactive. So let's see. Okay, so I'm going to try a live demo and see how this works. Huh? Oh, no, that should be okay. Should be here. Okay. Nope, that's not it. Yeah, I'm just waiting for it. So, okay, so you get to, was it F11 to make it full screen or something? Okay, all right, so here's the main page. So it just describes the project a little bit. So this is kind of the problem I'm talking about with all these plots. All these different, like you can run this in one line of R code and it'll give you all of the plots. So those are for alpha diversities, and then for the beta diversities, here the kind of like distance metrics that you can use to calculate, and there's quite a bit. So so you split it into multiple tabs. So these two, the ones that talk about alpha and beta diversity. Let's see. So here's a little bit. I'll get that to in a sec. And then the last two are just a little documentation on those. So so here's a little plot. So on the left, you can choose different data sets. These are just built in just for ease. And then below that, you can select, for now, just, was it 5 common diversity metrics that you might want to. So right now, this is just showing, oh yeah, I guess on the main part right here, you have on the top, you have the observed number of taxonomy or species or taxa that you're interested in. So these are just, per sample, you just look at the number of taxa that appear at least once. So the non-zero taxa there. So, and then the x-axis for the top one is just the different samples, and the y-axis is the diversity metric. The heat map is just to get like an overview of all the different samples at once, because the very top plot just shows you one, or it shows you the calculated measure. So the heat map kind of shows you for like each of the taxa. So the let's see, I think the abundance that is used to calculate those measures. So it's not really, I guess, to focus on too much, but just in case you want to get a global view of it. And then on the right side over here, you have a plot with this is per sample, so it's just one sample, and then the x-axis are the different taxa. So they have arbitrary names, so it's not too important to look at them. But the y-axis shows you their calculated abundance, or I guess their measured abundance. So some of them you can see appear quite a bit, and then a lot of them are a lot lower. So you can kind of click on these metrics, and they'll add new plots. You can add more of them as well, here and there. So it has that. This table summary is for at least just 2 metrics that we have. So the 2 columns that we have are different metrics. So I talked about some taxa that might occur once. So that's the singleton, and doubleton is the ones that occur twice. So that's just a sum of the number that occur those many times. What else? Quickly, so here the beta diversity. So just focusing on the top part above the this here. So each color is a different environment. So you can see that these are all, I guess, was it Euclidean PQA, or essentially PCA plots. It's kind of like this showing the differences between all these different groups. And so this bar here is sort of a normalization technique that you could use, and this will, I guess, change accordingly, and maybe you can see it'll update to show how to affect it. It'll take a little bit. We haven't really optimized this yet, so that's something we might have to look into. But if you take a sec, yeah, so update a little bit, and you can't see too many changes, but maybe with a more complex data set, it will change a little bit more. And the bottom 2 plots are just different, another, I guess, set of distance metrics as well. And these are the beta diversity ones, so these are the ones that you would use to compare environments. And then just wrapping up, so these are just a brief documentation of at least just 2 for now, because it took a little bit to format this, but for the alpha diversity, and kind of explain in more plain terms what these calculations doing. So like one of them, that's the math behind it, and it kind of describes what it does. And then similarly for the beta diversity. So I think that's it. Sorry if I took a little too much time, but yeah, so that's pretty much it.
What kind of data is input? Is it just counts, and like how are biologically, like what's the biological source of the data?
Yeah, it's mainly counts. So you have an abundance table, and what they are species. And
yeah,
so you can have like.
What I mean is like, where are you deriving counts from? Is it like a smear, and then, like, or is it sequencing based?
Yeah, so these are, at least for the built-in ones, I'm pretty sure they're more sequencing based, yeah. So like, what was it? So 2 of the data sets, like global patterns, huh? That one? All right, explain that one.
So for example, the global pattern, here you have samples of, feces. These are human samples. You have feces, swabs from the mouth and from the skin. They're bacteria. We're sequencing marker genes, and these marker genes tell you what species are present. And for each dot to see here is 1 sample or a swab. All right, and the colors indicate where they're derived from, either feces, skin, or tongue. Yeah, and the diversity metrics, they're calculating based on only the abundance of each species found in each sample.
Does that answer your question?
Yeah, that does.
Okay.
So it's like 18S.
16S.
Yeah, so we're back to it. Yeah, yeah. Yeah, and overlooked that stuff. Yeah.
Come on.
Cool.
You sure? I'm sure those graphs, you had no idea what they do. All right.
Oh, sorry. In the beta plots, what is the change? I've never.
Okay. So anyone that has worked with microbiome data or ecology data of bacteria, or with 16S, there's a normalization step where each sample, the library size, sample size is different. So you can't really compare with each other. You have to either scale it, which you're probably familiar with, or rarefy it, which is you make the sample size the same. The smallest sample size is about 100,000, which is all the way to the right. And sometimes in in pipelines, you may subsample to only 1,000 reads. Here, you can subsample to 100,000 reads. The smallest sample is
you're setting.
Yeah, you're setting.
Yeah.
That's great.
Yeah.
Yeah, so I guess it could be more clear instead of change count. But yeah, so it's a rare fact.
Certain studies is subsample to a very small number. So here you can see if you subsample to a very small number, how would the diversity change? How would the groups look with each other?
Mm.
Yeah.
Thanks.
Thank you for making that more obvious.
So it's really interesting stuff. I like this kind of diversity metrics, but how does something like this, where like, you're already making a stronger assumption about like classification of bacteria versus trying to figure out the level of diversity from just the level of diversity in your reads alone, right? Like, because I know that there are methods like you use like k-mers, right? Like the elements of different k-mers or whatever in the sequence. So like, does that type of data, is it as robust as then doing a classification and then doing these types of diversity measures? Or
you don't understand the question.
So you're saying like just use like some sort of statistical model to like classify these, or I guess I'm not understanding. Unless
I think it means like, for example, you can measure the OTUs versus the actual species. You think by species, so that's what you're doing.
Well, for
you think by OTUs, which is just text off units that are arbitrarily derived.
Well, this has nothing. This is after you've classified and you have the table. It can be species. It can be a table of phyla, table of OTUs, operational taxonomic units. These are actually, I believe, OTUs. It's an OTU table. We just call, most people here don't know what OTUs are, so we just call them species, and that's not technically correct. That's close enough for this audience.
Yeah. Does that make sense?
Yeah.
Okay. Yeah.
It's a responsibility to basically like sets of counts of something.
Yeah, we don't have that option right now, but we want to. That's the plan where you can input your own data and we can produce all these metrics and see if you change the sample size. We also have a function called sparsify. So in ecological data, in real ecological data, you have many zeros, or you have high abundance of some species, and most of them are very low. So just for the sake of seeing what happens, you can change that sparsification and see how that changes diversity and compare between groups.
This is a small question. So, we're gonna.
Okay. Yeah. Ask me afterwards.
Yeah, I see you guys talk afterwards.
Okay.
All right.
Cool.
Thank you very much.
I'm gonna change it. Okay. Yeah. And your slides? Just a link. Not Alt F4. To this? Yep. Cool. Alt F4.
All right, so I think I kind of, so our project was to develop tutorials for genomic analysis in R, and I think part of so this is a website we have up currently, and here we have currently the members of our team, and the list of tutorials. Not all of them are currently done, but we have put a few together. For instance, doing your analysis in R using reproducible pipelines. Here, for instance, is another tutorial showing how you can do genomic data analysis using large matrices that cannot fit in your memory of your machine, so you can store them onto your hard drive and on disk and load them as needed fairly efficiently using memory mapping. So I'm not going to go through the tutorials because I don't think that's the point of this talk. What I would rather focus on is the topic of our paper, which is almost done, written. We have a first draft going. Basically, hackathon-driven tutorial development. So here I'm going to just switch to a presentation. So first, I would like to thank all of the participants in my team. We were 5 of us here based in Canada, and we had a participant, Florian, joining us from Grenoble, France, so merci beaucoup. And we also had Yisong join us from Beijing, so xie xie. So how documented are your tools? Kieran and Rod kind of give a nice intro to the frustration that it can be whenever you're dealing with online tools, whether it's R packages or anything software related. There's different levels of documentation you'll find online. First level, no documentation at all, that's lame. Maybe second level is you have some documentation of like what inputs and outputs are expected, and there's obviously different degrees of detail that you can achieve with this. I think this is the bare minimum, just to describe what the inputs and the outputs. I remember coming across a software where basically the documentation is play around with the tool. And for instance, in this case, this is something that's required for CRAN submission, the reference manual, which is required for a package to be submitted to CRAN. One step further is something like vignettes, where you have like detailed, or well, I guess there's different degrees of details, use cases of how you can use a software tool. I think this is probably the most useful kind of documentation. I think everyone kind of learns really well by example. And this is, for instance, required for bioconductor package submissions. So every bioconductor package will have one or more vignettes demonstrating how the tool can be used. Again, I guess there's varying degrees of quality, but I think in large part that's what allowed bioconductor to be so successful and so accessible to the bioinformatics community. So our project goal was to organize our hard-fought collective knowledge into modular tutorials that use a common data set. The idea is that we all, over the course of our careers in bioinformatics, learn about different tools that aren't well documented and just eventually learn how to use them just by sheer determination. And to basically try and capture that precious knowledge into tutorials so that we can help train the next generation and probably the also current generation of bioinformaticians to do their analysis, their genomic data analysis more efficiently. So this is just a list of our tutorials. I already showed this, so I'll skip over this. I want to go over the benefits of hackathon-driven tutorial development. I think this was a first for me, and I think there's a lot of benefits from having doing it during a hackathon. I think it's motivating that everyone's working towards the same goal. Creating a tutorial on your own can be lame and annoying and boring. So just getting together as a group and working on a bunch of different topics together is, I find, motivating. You can cover obviously more ground as a group. You also benefit from immediate peer review. The fact that you can write a tutorial and then maybe two people write tutorials and then they swap and then you can review each other's work immediately. And then ultimately the tutorial will benefit and be improved from that immediate peer review. And probably the best thing about developing tutorials as part of a hackathon is, again, the collective knowledge you have as working as a group. You have different perspectives from different research groups, also from different regions in the world in our case. And so you're able to benefit from being able to distill that collective knowledge into something that is the most useful possible for a tutorial. And also because you're a group, you face a problem, you can problem solve much more readily because you're a group of smart people working towards the same goal. There are some challenges though, picking a focus. So the focus we picked was fairly broad, advanced genomic data analysis tutorials, and so you have to keep in mind what type of audience you want to target. A lot of tutorials out there, for instance, are introductory, and there's no shortage of them. And for instance, for this set of tutorials, I decided I wanted something more advanced because I feel there's a lack of that. So my audience is people who probably have a year's worth of experience in R, and that's just a decision you have to make. And yes, maybe people who are just starting off with R this might not be accessible for them, but that's a decision I took because there are a lot of people out there who have a lot of experience in R, but may not know these more advanced features or packages in R, and so helping them to get started is what my goal is. And also, you want to try and not reinvent the wheel. You want to make sure you look online. So bioconductor, I mentioned, is very good at creating vignettes. So maybe you don't need to recreate vignettes for those kinds of packages, but again, some of them might not have great vignettes, and so you can maybe build on top of their work and make even better vignettes, and maybe through your work you've learned new ways of dealing, new ways of visualizing, doing QC, whatever it is, you can basically build on the work of others and come up with even better tutorials. And what was key for us and what we spent quite a bit of time initially come across for our project was to make sure it's openly accessible, because obviously if you want a tutorial online and the first thing you need to do is get dbGaP permission, it's not going to happen. So something that's relatively tidy, so you don't spend much time tidying, unless that's the whole point of the tutorial. You want something that's interesting and relevant to your target audience. So in my case, like we were biased towards human data, which obviously made it more troublesome to find data sets that are openly accessible. Something that's multidimensional, so that allows for a huge number of types of analyses, and something that's not too small, so it's not too boring, but not too big, so that it doesn't take 24 hours to download. So trying to pick a data set that kind of fits all these criteria can be challenging. But once you identify one, then you can try and go all out with it and develop different types of tutorials for it. And just a few comments about virtual participation. So I think virtual participation, this was a first for me, and I think a first probably for hackseq, and there's a lot of benefits, I think, just from our experience in our team. It increases, obviously, the accessibility of our events. People don't have to travel from halfway across the world to join you. And we're kind of lucky with hackathons because they're particularly amenable for virtual participation. We have great tools like GitHub that allow for co-collaboration and task management. Slack was great for instance messaging. I mean, on the first day that we started, my 2 remote participants, one in France, which was 7 p.m., and the one in China joined us, and it was 1 a.m. in China, and we were all talking, and then eventually we did a Skype call that morning to kind of hash out the initial details of the project. This is maybe not realistic to expect that from all remote participants, but it's definitely doable. And for tutorials, you have the added benefit where you can each develop your own tutorial towards the same goal independently. So you're not like dependent on each other's work. So you kind of work in parallel, and then maybe at a certain point you decide to swap works and then kind of tweak each other's work. And so it's great for doing tutorials. Tutorial development is great, and it's for remote remote participation. Again, there are some drawbacks to participating in the socials. Sorry, Florian. And you also don't get access to the direct networking, although there is definitely some indirect networking. Obviously, we stayed in communication in the team, and I think that's beneficial. And the key thing about whenever you have remote participants, which might be struggle for, let's say, team leads, is to make sure that there's a constant and clear communication to make sure everyone at any given time has a task. So I guess that's a wrap for hackseq 2017 for us, and I'm already looking forward to hackseq 2018. Hint, hint.
Well, yeah, so I think you identified a really key spot, key challenge. But I guess, so like people who come to the hackathon with a key number of elements and how do you say what they want, so how do you think you can kind of like evaluate whether you're pitching it correctly? Really, how do you get feedback? Aren't just experience in trying to
So are you referring to the people who are eventually going to use the tutorials, or the people developing?
But I guess that's just so people who develop these tutorials are all and talk to each other, all are key areas. Like how do you make sure it's actually accessible to accessible?
So yeah, that's definitely that's a challenge. I think maintaining your audience. Well, yeah, first picking an audience and then maintaining that in mind when you're developing tutorials is important. And tutorial development isn't like trivial. It requires some I guess. You have to realize that not everyone has this level of knowledge that you have, and making sure that you describe why you do each step in way that is accessible is definitely important. So there's definitely ways for an advanced user to come and create a tutorial that is just not accessible to the target audience, and that's something that you have to be careful of, because otherwise you're wasting time creating a tutorial that a small fraction of people can benefit from. So I guess that's why, like, initially you really have to pick your audience. Is it meant for anyone who's starting off with R? Is it meant for someone who has 6 months, a year worth of experience in R? And try and tailor your tutorial for that audience. And also make it clear from the outset okay, this, I expect you know this, and maybe point to other resources. Again, there's tons of resources online, especially for on the introductory side of things. If let's say, okay, I expect you to be familiar with genomic ranges before tackling maybe this more advanced package, well then you would like, okay, go to this vignette first and then tackle this tutorial. So that's definitely something you have to be careful of, because otherwise, yeah, it will fall flat.
Cool. Team 6. Santa. Oh yeah.
No, sorry, one further reference projects. Yeah, OQC.
Yeah. Cool.
Come on.
We're Team 6, Chrome QC, and our project was to develop a quality control metric tool for 10x Genomics Chromium linked read libraries. We had a relatively big team, and this project was a very serial pipeline. Like each step depended on the step before it, and fed into the step after it. So my primary purpose was just to decompose this pipeline into individual stages, and then each person volunteered to implement one of the stages. And that's how we structured our project. So let's introduce my team. We got Justin, Bada, myself, Sean, another Sean. I finally cloned myself. I'm gonna be so much more productive now. Emre. And then we had 2 on-site teammates who aren't here today, Tanya and Hamid. And we had 2 off-site teammates, Ryan in California, who's actually a 10x employee, and also Swapna in India, who was 13.5 hours ahead of us. Also Chen. Oh, and Chen. This is why I told you to stand up here.
Chen drew that.
And Chen, yeah. And Chen drew the logo, which I love. So I'll just pass it off to my teammates to present our tool.
The idea is that we are targeting 10x Chromium long-range reads, and the idea is, well, you have a data set, a sequencing experiment, and you want to just quickly check the quality of your library preparation and the success for that. And that is mainly can be seen by looking at the molecule sizes that you can infer from your sequencing data and the barcode information that you have here. This is already done by Loop, which is a software package that 10x Genomics provides. It's a cool tool, it's a great tool, but the problem is that it does way more than you actually just need to just quality control the data. It tells you SNPs, it tells you structural variants, it does a lot of things, but then it also takes a lot of time. So then you have something else that we are also motivated by, which is FastQC and MultiQC, which are these very cool and nice tools that look at your FastQ files and they report simple metrics on base qualities, and MultiQC aggregates multiple FastQC reports together, but they don't have any metrics for long-range reads, such as Chromium reads. So our goal here is that we wanted to build something that is simple and quick as FastQC that can be aggregated into multiple report files, while at the same time providing the basic quality information that we need for long-range information.
Yeah. All right, and then so we're all bioinformaticians here. We're a lot like old workers. All we do is build pipelines all day. So then, like was said previously, Long Ranger, right? Long Ranger performs their analytics on the entire set of reads. This is because the depth of coverage is very high. You can still get a good report, a good summary of the your library preparation with a subset of the read. So what we do in that vein is to sample the barcodes randomly, so then we get a good coverage over all the molecules. And then given those barcodes, we then extract the reads, we align the reads onto the human genome, or the genome that we're looking at, using Minimap. And then from there, we group reads into molecules based on a simple heuristic. If the reads are greater than 60 base pairs away, we can assume that they're not part of the same molecule. And if they're close enough, like within 60 base pairs, then they're within the same molecule. Kilobase pairs. Oh, kilobase pairs. Yeah. So. KB, yes, that's more reasonable. Okay, yeah. And then so step 4, which is the most essential step, is that we estimate the size of the molecules. So what Long Ranger does is that they take the complete set of reads, they take the read that maps the leftmost, and then they take the read that maps the rightmost, and using those two numbers, they infer what the molecule size is. So that's statistically a good way to do it if you have a large data set. And then they don't do any statistical stuff. They just take the leftmost and the rightmost position to be the estimated length of the molecule. Well, minimize it. And then but what we do is that we use a maximum likelihood estimator where we assume that the underlying distribution of the mapped reads is a uniform distribution. And using a unbiased maximum likelihood estimator, we infer the start and end position of the molecule just based on the reads that do map, and using only a subset. This is a really efficient way of doing so. And as we'll show soon, that this is an accurate estimation of the length of the molecule. So we get very similar results to Long Ranger with a smaller data set. And then from there, we perform other statistics on the molecules, just as Long Ranger does, and then we generate a report. And then over multiple reports, let's say you have multiple runs, and you want to see how good your library preparation is over all those runs, we use multi-QC to aggregate them and then to make a final report. And then so some demonstration, I guess we have
So this is the multi-QC. Oh, this is okay. So yeah, we'll have a multi-QC output as well. That wasn't quite ready yet, but
No, it's ready.
we have that. You have a link for it? Yeah. Okay. Then Chen should be presenting that. Anyway. We totally prepared this beforehand, guys. This is one of the outputs. Swapna actually worked on the actual way this report looks. It's based off of our Flex dashboard. So I'm sure you recognize this ggplot plot. So the main output of our pipeline is this, and it actually uses a corrected distance to estimate the, like was mentioned before, using maximum likelihood estimator. And it actually, using only a subset of the data, it actually recapitulates what we obtained with Long Ranger, and that didn't work. Essentially the mean molecule size is what we'd expect, and the distribution looks pretty much like was shown before. In addition to that, we actually have other metrics that that might be helpful for people who want to know how well the library was constructed. I mean, you could run FastQC, but this is essentially more information about the actual barcode information, about like how many molecules you have and how many barcodes you see with each molecule. Though we have to probably adjust it for the fact that we do sampling, and that's still something we need to test. So that's pretty much it for the output. The thing is that from here we could actually derive many more statistics that we have from the entire pipeline. So it's only kind of the beginning of what we can report.
Can you click on that multi-QC?
Is there one?
Yeah, it's back to the slides.
Oh, okay. Sorry, I thought that was like a caption. Okay, got it.
You do.
Okay, so I didn't prepare for this, but I'll try my best. So yeah, multi-QC is a tool that can actually automatically find a bunch of your TSE files and aggregate across multiple experiments. So in this case, there's actually 3 experiments, or 3 different sequencing experiments, just showing you the relative molecule size of each one of these. So this is the Ashkenazi trio. So essentially, it shows you that they're all pretty much very similar. I mean, some of them have many more have possibly a better distribution of molecule lengths. Like you probably want longer molecule lengths, so it's possible that this library is the kind of the best of that. But as a whole, the structure is what you'd expect. There's it's a unimodal distribution, and we hope that we can expand this to more of the information that we get from the other report. Yeah, that's it. There's also way more that we can do with this, but all of the building blocks are there for us to generate way more information quickly within like, an hour, less than an hour, probably like 30 minutes or something, as opposed to a long ranges, which takes like 3 hours, something like that. And yeah, so that is the end of that. Where's the? Thank you. Any questions?
Hey, Rob. So do you have a sense of like how little I guess, yeah, I almost feel like pulled in two directions. It's like how little can you subset to get an accurate report? And or would it just be worth like having the full report run and putting that in?
I think you can do them both. Like you can start the full report, and you'll probably do that anyway, right? And while that's running, you can run a quick report and get a subsample report. And how far can you subsample? We haven't done that testing yet. It's something I do want to do. But my thinking is that we can use about 1/10 of a lane in terms of reads, and then but within that 1/10, we only have to look at about 1/1000th barcodes. Because there's a million barcodes, and 1,000 should give you a good representative sample.
Kind of like where you like first build and say, okay, stop, and like do something else, or is it kind of like once you've done it, and then you need to start that?
I suppose we should have had a slide on how 10x Chromium works.
Oh, yeah.
But essentially, no, it's a limit of sequencing with a special library preparation stage where you have reads that are linked together with a barcode, almost like MapePair data, except you have multiple reads that are all associated and across a longer distance potentially.
But answer whether, like, once you've prepped the library, sequence one lane, and now you can choose whether you can sequence more lanes, or if you've done multiple libraries, which libraries you want to sequence more of.
And considering statistics, the maximum likelihood estimator is a function of n, where n is in the denominator of a correction term. So as you increase n, your error reduces by a factor of a quadratic factor. Yeah, so then it decreases quickly as you increase n. So then maybe like the 1/10, you may 1/2 if you're very stringent, they'll give you a good estimation just given based on the estimator.
Right, thank you guys.
T8. It's close.
Yeah, go for it. Hey everyone. So we worked on trying to develop a way of measuring the efficiency of differently designed sgRNAs in CRISPR. So for those of you who don't know, and I was one of those, you use a little bit of sgRNA to actually point to where you want to do the cut or mark or anything like that. But there are different thermodynamic properties of these sgRNAs that can get in the way of marking what you need to mark. And in addition to, if you try to cut, let's say like the 98th part of a peptide, there's a pretty good chance that it actually might not do anything. So I would like to thank my team for helping, well, make this all possible. We have Inez here, Sasha, Jessica, and Pranav. And then we also have Sinchan, who was working from India. And by the way, hi, I'm Matt. So the data that we had was for C. elegans, and we had a total of 75 reads. So we did a lot of data cleaning. But what we ended up doing was we took another data set from the Donch lab, who originally wrote this rule set. We looked through their methodology for creating essentially these machine learning models. and discovered that they had actually not done any hyperparameter search. So there was actually a lot of movement inside of there where we could improve the model for both human and mice, as well as trying to apply this technique over to C. elegans. So we had a lot cut out for us, so we had to use as many different strategies as possible. One of them was simulated data, which Inez very graciously did. We haven't used it yet. We're looking at possibly using this for some sort of semi-supervised approach. As they say in data science, it's 80% cleaning the data and then 20% complaining about cleaning the data. So yes, it was a lot of cleaning the data, and that was everyone on the team. I'm very grateful to them. We also looked through the literature that was written. The original paper was written in 2016, which is forever ago. So there were some improvements, and we took as many improvements as possible. Also looked at different future directions and tried to apply that. Some examples were the folding energies of this sgRNAs, and there are some 3' motifs that were considered especially important. The last thing that we did was hyperparameter search, and since this is bioinformatics, I thought it would be funny to do evolutionary algorithm, and that actually worked out really great. So yeah, this is how machine learning works. You just pile everything into a big matrix, do some linear algebra, and then something comes out, hopefully. And it did, it totally did, it worked. So right here is the base. So this was the original library. And essentially what they had done was they just took a gradient boosted tree regressor with no hyperparameter tuning at all, and they got a pretty decent result. But it turned out we could do a lot better by adding features. We also just did the classic thing that you do when you don't have any ideas. You just put an XGBoost, which is another algorithm that's very common. So we tried that right there. But then we used this evolutionary algorithm, and you can see as the generations go down and down, it actually outperforms the XGBoost, still within the significant, I should say, this is cross-validation between 5 different folds. So we could actually reduce down these confidence intervals a lot more if we increase the level of folds. But that's basically where we are. We attempted to do this on the C. elegans data set, but the data is just too dirty. We don't have enough reads. So despite valiant efforts, we're gonna have to save that for another day. But we do have an improvement over the baseline set. So yeah, that's everything.
So why did you choose this project if you kind of believed in not really knowing a lot about using witness numbers?
Yeah, so I am not a bioinformatician. I'm an imposter. I'm a data scientist though, and this project was actually proposed to the UBC data science graduate team, but being the only one of 3 people who had a biological background in that program, this never got off the ground for us. So I wanted to take a crack at it and see if there was anything to be done. So,
what is the evolution one?
yeah, actually, this is really interesting. I should have talked a lot more about that because I think this is really fascinating. So I'm just going to talk until you guys kick me out. So there is a package in Python called tpot, which is like tree something something optimization something. Essentially, what it does is you start out. I'm going to say with the base parameters here. You take 10 models, just randomly chosen. They're not going to be good at all. But what you do is you take the 2 best of those models based off of cross-validation. You throw out the rest. And then from those 2, you make 5 children. And the way that you make 5 children is you just do a single point mutation to the model. So that can include changing a hyperparameter, changing the model, or also adding like a preprocessor of some kind. So what's really great about tpot over other like auto machine learning type algorithms is it evolves an entire pipeline instead of just doing hyperparameter search on the single single model. So we were getting a lot of like really exotic ones. If I remember correctly, this one right here is, sorry? No, that was for C. elegans. What was weird was we were getting like a lot of things I did not expect in C. elegans, but in this one it actually was pretty well behaved because we were getting a ton of very heavily regularized like regression, but those regressions were actually feeding into different models. So they would use, say, like a regression, take the output of that, do some other like normalization onto it, and then use that as an additional feature for like a larger regression, or sometimes it was also like a random forest type model. Yeah, it was really weird. I should have actually just shown what some of the things that were being evolved out. But yeah, there's multiple layers deep. Any other questions?
Is there a reason why you did this grid search or something?
Yeah, so grid search, in that if we set up the grid search on hyperparameters or even did something smarter, like HyperOpt is a library for this, we would just stay within the single model and we would get pretty good, right? But because we only had essentially like 48 hours, I needed something that I could just like run overnight and see if we got any improvements. And like, that's just how it worked out. And also it seemed really cool. So I wanted to try it out.
Cool.
Yeah.
Okay, last, and certainly not least.
All right. So we're team 9. We worked on trying to collapse a phylogenetic tree using taxonomic metadata. We started out thinking that we could use this existing function within the PyTools package called collapse tree. It worked by graphically showing you the tree, and then you could click on it with the mouse, and it would measure the distance between the mouse click and every node in the tree, and it would decide which node you clicked on, and then collapse that node. When it collapsed that node, it used a subfunction called split tree, which we ended up using. Lauren hacked her way through split tree and made it so that we could use it. In my lab, we use really big phylogenetic trees sometimes for our diversity analyses. And when I make the tree, I can't really evaluate it. It's too big to graphically represent in a way that you can just look at it and say, is this tree okay? Are there large-scale problems, or are things falling where they should fall within the phylogeny? You have R packages that exist that allow for visualization of tree data, but none of them are really good for huge trees with thousands of nodes or tips. So our goal was to use metadata with taxonomic strings to collapse the nodes within the tree to a specific taxonomic level and then visualize that. The metadata is pretty simple. In the columns, you have taxonomic ranks, and in the rows, you have taxa. So the user supplies the metadata. It's like I just described, a tree in phyllo format and a rank to collapse. Oh, did we even say this is an R? Yes, this is an R. A rank that they want to collapse to. So some people label their ranks by like kingdom, phylum, class, etc. But you could label them anything, 1 through N. And an optional list of clades that you want to exclude from the collapse function. You can actually, we allowed the users to pass either a character vector of N clades that they could type out, or a column in the metadata that marks which clades they want to exclude. And the output is a prune tree where the collapsed clades are represented by a new tree tip with the clade
name as the label. It looks like this. So this tree here has about 200 tips, I think. Look at it, and if you spend some time with it, you understand what's going on. But you could apply aesthetics to it, like coloring by clay that the tip belongs to. But our thing really reduces the number of tree tips, and everything that you don't care about, like if you want to look at a couple of clades, you just exclude them. In more detail, I mean, you just exclude them from the collapse function, and they show up like this one here. So this is the, what was it? Green plant. So this is a real subset. And then this one was polychaete worms. I like it. It makes the tree easier to read. I don't know. If you wanted to do something like this, you could do something with a visualization software, but the computation time is pretty high. Like it takes a long time to generate a figure of a tree with a lot of nodes or a lot of tips. I mean, I keep saying nodes, but I mean tips. We didn't escape that problem. We thought we fixed it yesterday. It worked really fast, and then we fixed a bug. And today, if we used a tree with 3,300 tips, and one iteration through a clade took 15 minutes. So it takes too long still. But with this tree with 200 tips, not so bad. It takes about, I don't know, 5 minutes. So, so future goal, we want to add this function to the PhiTools package developed by Liam Ravel, if he'll have us. We created this documentation file. Mark did the documentation for us. Problems in roadblocks. The structure of trees in ours is real basic. There's a matrix of edges, a vector of tip names, and a vector of edge lengths. The internal nodes don't have labels, and it makes it difficult to track them as the trees change. Lauren should talk about this part.
Oh, sure. So basically what we thought we had coded yesterday, and we were all excited that it was working, is, this is kind of hard to explain. Let's just like gloss over it. But essentially, if you have nodes in a tree, they're just kind of labeled based on indices, where the tips are the first indices, and then all the internal nodes are labeled after that. So we were trying to increase the efficiency by determining which nodes to collapse before we started collapsing, so that then you could just cut them all out at once and not have to do it tip by tip. But the problem is once you do that, then all the indices change. So what used to be the node 11 that you were going to get rid of here is now relabeled as node something else, and there's pretty much no way to track it based on the way that it's written. So when we realized that this was happening, we kind of had to try and use some like tool that tries to compare the two trees and matches them based on the names of the nodes, so that it would tell you that this one corresponds to this. But that's what takes basically forever.
Comparing the trees takes way too long.
Yeah. And then the other problem was just that everything is very poorly documented. I think probably the people that write the majority of these phylogenetic visualization tools are biologists, which makes sense. And that's why they're useful. But
just like us.
Yeah, no, I mean, it makes sense that they would write the tools because they're the ones who are going to be using them. But the code, probably like our code, is not particularly well documented.
So we were working on this today on adding parallelization to the tree comparison, like the match tree function. So getting the new node IDs takes a lot less time. We're a little ways there. We reduced it from 20 minutes to less.
No, it's about 5.
5 minutes? Yeah, okay, so we're getting there. So 5 minutes in iteration, pretty good. There's just like 122 of them.
we want to, let's see,
Oh yeah, this is.
consider coming up with our own tree structure.
Yeah, so
Go
one
ahead.
of the things that I was looking through this, trying to figure out the best way to split a tree, because I decided the function was a little too clunky. But then I realized that the entire philo object represents the tree as a bunch of edges, and therefore the problem is if I clip off an internal node, the ideal thing would be to drop all children nodes automatically. Instead, when I clip off an internal node, I have to go through one by one and clip out every single one of its children because it's a list of edges. So the best possible thing would be to have the tree represented as some sort of linked list where the parents and children have pointers to each other. That way, if I snip off a parent, then all his children and all his descendants just get destroyed or tucked away somewhere, which is a lot better. Which is one of the things why Lauren was saying that this probably was written by biologists. They don't, because sometimes when you have certain data structures and there are multiple data structures, you should choose the one that's the best representation for what you're doing. If you want to be cutting trees, pruning them, manipulating them, I don't think the
File class.
yeah, I don't think the file class is actually the best decision, but I guess they definitely consider this when they're doing the implementation.
Mhm. And I don't know, we have a little wish list, but that's for later. Thanks to my team. They're great. Questions?
Yeah, so I think this is really cool work. I guess I have kind of a broader question. So
Mhm.
as you're saying, like people are probably doing trees have been around for a long time. But like as you get to thousands and tens of thousands of things, it's almost like the visual metaphor starts to break down, and like kind of the analytical metaphor almost kind of starts to break down. So when you're doing like gene expression analysis or something like that, there's still a tree there, but you might think of things just in terms of like a distance matrix or something like that. So do you think we're kind of reaching kind of the point where you move away from the literal tree to just distances between everything and do like clustering and like that set analysis, or do you still think like we're going to be making bigger and bigger trees for the next 10 years?
Oh boy, I don't know. I'd like to see somebody compare the two. I haven't. Yeah, that'd be interesting reading, but I don't know. Probably like the bigger the tree gets, the harder it is to understand. So, yeah, like I don't expect to look at a distance matrix with my eyes, but I can still use it. Yeah.
I feel like the most important part of phylogeny tree and the reason why it was created in the first place was for easy visualization. So I feel like if your tree gets too big to visualize, that you kind of defeat the purpose of using the tree in the first place. So yeah, I definitely think that having efficient ways to visualize big trees is practically vital. But if you can't do that, you might as well just go back to a distance matrix. That's about how much information you can get from the picture.
Mm-hmm. Anybody else?
Well, thank you guys again. So before you head off, and it's going to be something that will take just 5 minutes. We're going to vote for People's Choice Award. So I'm going to drop a link onto the Slack channel, so people might have to get their laptops out. And virtual people, if you're there as well, please vote. This is an honor system. Please only vote once. Oh yeah, and also please vote for another team. Or if you really are so determined that your team was the best. Let me just pull the link for this, sorry. Oh. I'll put it in the general. Just in case there are any virtual people that have already logged out.
I want to see votes for.
And vote for a different team. Cool. And we'll give people 5 minutes in case virtual. people need a minute. So feel free to talk amongst yourselves, or if you have any burning questions with another team, now is the time to go hassle them. And organizers can chat to you down in the corner. And someone bring the laptop.
Someone was supposed to take that out and didn't.
Can you grab your laptop?
Yeah, we should for sure representation. Right. I mean, there is the question of perspective process. Yeah, I mean, perspective process the two components. Yeah. Like right now, it is because they're one. Yeah, exactly. But if you feel like then it would be functional. Like each group is yeah. Oh, that is yeah. Okay. And like those 50% of my group. Yeah, exactly. But I explained that she's going to portion. Yeah. Like up for 3%. Like oh, seriously, yeah. So deep fried. Or it could be like deep fried, but I'm open to Maybe have the same amount of real that we have.
Okay, another 2 minutes. If you haven't already. So, any virtual participants, vote now. Another minute, 2 minutes.
I mean, it's like a actually, stewardship organization. And those would be virtual documents, just one.
You guys, we do want to kind of quickly clean out.
All right. I think.
About to all over myself.
Yes. Just stand in front of this.
Okay, guys. So our poll is closed. So I can give you the results. So we're gonna have some gift cards for the second and first place teams. So in second place is Team 3. Congratulations. Wait to make, oh, we don't have the gift cards. They're gonna be electronic because you guys have virtual participants as well. But you can, what? Oh yeah, take a picture. Come up here. Maybe we can put your presentation slide up.
I had a speech and everything.
Do the speech while I pull up your presentation. I'll pull this up and we can take a picture of you with this on the screen. How do you full screen?
Present.
It doesn't know how to work a camera.
Wait, I got it. I thought
Jake? Okay. Does she have it? Did you get it?
I'll describe it.
Yeah, Bruno, do backup photos.
Nice. Yeah.
Okay. Sorry, it wasn't my best idea. And the winners are Project 1, Biosyntax. Congratulations. Oh no, yours is a file. Oh, here it is.
Yeah,
Great job, guys.
5.
No, I know how to do this one.
You're like,
Wait, but we need like Alyssa and Eric, so we can put some space.
you don't Photoshop the thing, that's good. All right. Well done, guys.
Congratulations, guys.
All right, so this is the end. Thank you very much for all your incredible work, and thank you to all the people around the world that have also been working hard on this. I hope you go to sleep now, wherever you are. And basically, you're all fantastic, wonderful people, great scientists, and there's a ton of food out there. Please take some of it, if there's any left. Chantel's been protecting it, ish, so it's probably not been just like lifted. But yeah, you guys are awesome. Bye.
Thank you, organizers.
Here we go.